Hello dear students, welcome to Biology Decrypted. In this series of Biochemistry Decrypted, we have covered multiple concepts so far. We have discussed about the structures of amino acids. Well, if you haven't watched the previous videos, all these videos have been clubbed as a playlist and the link to the playlist is available on the I button above. Do watch all the lectures and especially the previous lectures before coming to this lecture because all the lectures are linked. There are essential concepts which you need to know. Only then you will be able to connect and you'll be able to understand better. Well, without much further delay, let's start lecture 6, that is amino acid titration and charge on a peptide. Well, these topics are very important from examinations point of view. So please watch the video till the end. Now let's start. Let's talk about amino acids and let's revise all the structures of amino acids which you had learned in the previous lecture. I hope now you can draw all these structures on your own, and you can also tell me the name of these amino acids just after having a look. So which amino acid is this one? This is glycine, G. Then this is alanine. So glycine, alanine, valine, leucine, isoleucine, this is methionine, this is tryptophan, this is phenylalanine, and this is proline. So Gavli, MWFP, these are all the non-polar amino acids. As you can see here, the R groups here, the R groups in all of these cases, these R groups are non-ionizable, which means they do not have any such group which can be ionized. They only have two groups that can be ionized, that is obviously the alpha carboxyl group and the alpha amino group. So these are the only two groups that can be ionized in this case. Now coming next to the polar uncharged amino acids. Well, if we talk about the polar uncharged amino acids, this is, as you can see, serine, this is threonine, this is cysteine, this is tyrosine, this is asparagine, and this is glutamine. I hope you can recognize these amino acids by their structures. You can also draw their structures. So if you can't, well, the previous video will help you do so. Now, coming to charged amino acids. If you talk about charged amino acids and also specifically about acidic amino acids, we can see here there are two acidic amino acids. The first one is aspartate or aspartic acid, and the second one here is glutamate or glutamic acid. Now, these are the charged amino acids with acidic groups. Now, what you can see here is there are three ionizable groups here. All these three groups are ionizable. Similarly here, all these three groups are ionizable. In case of polar uncharged, well, what we can see here is in this case, in this case, in this case, and this case. Well, these groups are not ionizable, but what happens in case of a sulfhydryl group is sulfhydryl can still lose its proton and get converted into this ion. Similarly, if you see here, in case of tyrosine, this is a phenolic group, and phenols are acidic. They are acidic like carboxylic acids. So they also behave as weak acids, and yes, they lose H+. So, well, this can also get ionized. So in this case and this case, there are three ionizable groups. For polar uncharged amino acids, well, for most of the polar uncharged amino acids, there are only two groups, but only for tyrosine and cysteine, we can say that there are three ionizable groups. For acidic, we have already seen. Now coming to the last part, that is basic. Charged amino acids, basic. In this, you will be seeing there are three important amino acids. First one is arginine, the second one is lysine, and the third one is histidine. So R, K, and H. And here, as you can see, here there are three ionizable groups as well. So here there are three ionizable groups. So now we saw the structures. We actually revisited the structures of all the 20 amino acids, and now we can say that few amino acids, that is mostly the non-polar ones, all the non-polar amino acids, and most of the polar uncharged amino acids, they only have two ionizable groups, that is the alpha amino group and the alpha carboxyl group. Whereas if we talk about the charged amino acids, that is basic and acidic, there are 3 ionizable groups. And if we talk about 2 special cases, that is of cysteine and tyrosine in case of polar uncharged amino acids, we can see that there are also 3 ionizable groups. Now let's come to the general structure. Let's talk about the general structure of amino acids. Let's talk about the ionizable groups in general. So let's talk about the ionizable groups in case of amino acids. Well, for amino acids, there are ionizable groups that are actually common to all. So ionizable groups that are common to all are obviously the alpha carboxylic acid group and the alpha amino group. The alpha amino group. In both these cases, what I have drawn is the protonated form of both these functional groups. So both are there in their respective protonated form. Now what's important to see here is the net charge. What you can see here, the net charge here is 0, whereas if we see the net charge here, what we see here is the net charge here is +1. So we saw the net charge in both the cases. Now if we say, if both of these lose their protons, for example, if alpha carboxylic acid group is losing its proton, so obviously losing a proton, this process is called as deprotonation. And in both the cases we are talking about deprotonation. So what we are getting here is, in this case we are getting alpha carboxylate. We are getting alpha carboxylate, and obviously this is drawn in this way, COO minus, like this. And similarly, if NH3 plus is losing a proton, this is your alpha amino group with its intact lone pair. So now if we again measure the value of net charge, what we get here is something like this. Here the net charge in case of carboxylate ion, it is -1, and here if you see in case of amino, alpha amino group, from +1 it is becoming 0. From +1 it is becoming 0. Now if we try to draw a general structure of amino acid like this, we are drawing the Fisher projection. Here we have H, here we have NH3 plus, here we have COOH, and here we have R. Now we are considering that R here is non-polar. R here is non-polar. So what we can say is this alpha amino group, and obviously this is the alpha carbon, this alpha amino group and this alpha carboxylic acid group, both these groups are the ionizable groups that are common to all amino acids. This will be there in all the amino acids, and this will be ionized in all the amino acids, irrespective of the side chain, that is R. Now let's talk about the concept of ionization further in detail. So what we'll do here is we'll draw the general structure again, COOH, NH3 plus, H and R, and let's say we are considering specifically non-polar and most polar uncharged amino acids. So here what we'll see here is this one will get ionized, this will lose a proton, and this will also lose a proton. Fine. So these two groups will get ionized for sure, and these we'll be looking at each of these groups separately, like not in this particular amino acid but separately, So let's first look at the case of ionization of alpha carboxylic acid group. So this is the first ionization that we'll be seeing, that is alpha carboxylic acid. Obviously this is an acid. This is getting ionized, and what is it forming? It is forming its conjugate base, that is COO minus alpha carboxylate, and this is the conjugate base. Along with it, it is also forming a conjugate acid, that is H plus ion itself. So this is the conjugate acid. Now if you try to write the Ka, that is the dissociation constant, we can write alpha carboxylate into H plus ion concentration divided by alpha carboxylic acid. This is how we write the Ka expression. And here, since this is the first ionization that is taking place, first dissociation that is taking place, so this will be Ka1. And for Ka1, if we take the negative log of Ka1, we get pKa1. So this is the concept of pKa1, or it can be simply called as pK1. Some books simply refer to it as pK1. So this is the first ionization taking place. The carboxylic acid group is getting ionized. Next, what will happen is, the next ionization that will take place is of alpha amino group. Now this is already protonated, so it's behaving as an acid. It is getting dissociated. Alpha amino group, this is the conjugate base, and along with it, it is losing a proton, which is the conjugate acid. Now again, we can write Ka2 here. We can write Ka2. Ka2 will be equal to alpha NH3 plus concentration into H plus ion concentration. Sorry, this is alpha NH2 concentration multiplied by H plus ion concentration divided by alpha NH3 plus concentration. This is how we will represent it. And again, if there is Ka1, obviously taking the negative log, we can get, if there is Ka2, we can obviously get, by taking negative log of Ka2, we can get pKa2, or simply we can say this is pK2. We can simply say this is pK2. So this is how we get pK1 and pK2, and this is common for all. So the ionization of this will be associated with pK2, and this will be associated with pKa1. Now let's further understand the mechanism. What is happening actually? In the first ionization, what is happening is, basically we are carrying out titration. Since we are carrying out titration, we will be titrating it with OH minus ions. Well, why? Because OH minus ions will try to abstract the protons. They will try to actually kidnap away the protons, and the one that loses easily will actually lose the protons at a lower value of pH. And similarly, if there is a group that is folding on strongly to the proton, it will be requiring a higher pH or a higher OH minus ion concentration. Now, as we know, pH is equal to negative log of H plus ion concentration. So what we can say is pH is actually inversely proportional to H plus ion concentration. This is what we can conclude. And also what we can say is, this is inversely proportional to acidity. This is inversely proportional to acidity, as H plus ion, increased H plus ion concentration, they correlate to acidity. So OH minus ion concentration, it is actually directly proportional to pH. Very, very important. If we are increasing OH minus ion concentration, we are also increasing the pH. Because in this way, pOH will be inversely proportional to OH minus ion concentration. But this OH minus ion concentration, what we can say is, this and this, that is pH and OH minus ion concentration, will be directly related. This is how we can actually we can conclude this, that OH minus ion concentration will be directly proportional to pH. and obviously inversely proportional to POH. Now, let's consider the ionization of carboxylic acid group. And what we generally do is we actually take this particular acid, we finally ionize it, and after ionizing we are getting a conjugate base. Now we look at the stability of the conjugate base. If the conjugate base is stable, if actually the lone pairs, because the basic role of a base according to Lewis theory is to donate a lone pair. Now if the base is not, if it is not doing its duty properly, well then it will be a weak base. And if a base is weak, if a conjugate base is weak, then obviously the conjugate acid will be stronger and it will be dissociating earlier. So talking about the conjugate base, that is carboxylate COO minus, it is COO minus. Now what will happen is there is a scope of resonance. There is a scope of resonance here. This electron will become bond, this electron will become bond, and this bond will become electron, and as a result you will get this carboxylate ion. Now obviously you will get a resonance structure like this, COO minus, and here you will get C double bond O. So this is resonance stabilized. This is resonance stabilized. And now what you can see here is this particular conjugate base. This conjugate base here is a weak base. This is a weak conjugate base. And since the conjugate base is weak, and also what we can write here, these are the resonance structures. These are the resonance structures or the canonical forms. So we can say these are resonance structures, and we can actually have a resonance hybrid from these structures. So what we can say here is this is the skeleton, and what we can see here is the negative charge is actually delocalizing between these two centers. So this is the overall structure of carboxylate ion that we can make. And what we can see here is these lone pairs are actually not staying on one oxygen, rather it is being delocalized. And this is how this particular ion is getting resonance stabilized. So this is stabilized by resonance. This is the resonance hybrid. Now what we can see here is that the H plus ion here, obviously the oxygen won't be binding to hydrogen in a very strong manner. It will be losing it very easily. This will be easily deprotonated. This is very important to understand. And what we see actually on an average, the pKa1, that is the pKa for this particular group is actually on an average around 2.3. So what happens here is a very simple reaction. Basically, this is your carboxylic acid group. Now what happens here, there is your OH minus ion concentration while you are titrating. This will make an attack here, and this will try to abstract the proton, and the proton will give away the electrons to oxygen, and what will form here is COO minus, and this will form H2O. This will get lost as H2O. And this is how carboxylate ion is formed, and this is the overall deprotonation. This is the process of deprotonation during titration. This is how the proton is actually lost. You can see here, this is how the proton is actually lost, and it goes out as water, and it leaves behind the carboxylate ion. Now if we talk about the second ionization, number 2, that is the ionization of NH3 plus group. So this nitrogen is already bonded to a hydrogen, and it has already donated its lone pair via a coordinate covalent bond, and because of that it is having a positive charge on it. Now what is happening here is this OH minus ion is coming. This is again trying to abstract this H plus. This H plus is giving its electron back to nitrogen, and as a result, what we can see here is NH2 group is formed. NH2 group is formed, and obviously this is conjugate base. And here there is no resonance. Here there is no resonance. Here there was resonance, and obviously this lone pair was not available for donation. So it was not behaving as a strong base. It was rather behaving as a weak base. But what we can see here is this is a strong conjugate base. And strong conjugate bases have weak conjugate acids, and therefore this ionization requires a higher value of pH, or you can say a higher concentration of OH minus ions for ionization. And therefore what we can say here is for this the pKa value, that is pKa2, is approximately 9.6 to 9.7. It is approximately from 9.6 to 9.7. Now we have understood the mechanism, the overall concept of ionization. Now let's come to the actual part of titration of amino acids. So we'll be talking about titration of amino acid with non-ionizable R group. R group. This is what we have been talking about. Well, which amino acids will be coming under this category? Gavli, MWFP, obviously, because these are all the non-polar amino acids. And also S, T, N and Q. S, T, N, Q, and this will be from the polar uncharged category. These will be from the polar uncharged category. Now, let's take an example of alanine here. Let's take an example of alanine. Alanine, that is A. This is the structure of alanine, COOH. Here we have NH3 plus, H and CH3. The R group here is CH3 group. Now, if we talk about the titration, well, for titration we'll have pH on the y-axis, and we'll have equivalents of OH minus ions added, that is the OH minus ion concentration, on the x-axis, and we'll be adding OH minus and we'll look at the pH. So this is how it will change, and then there will be one ionization, there will be another ionization, and this is how this whole graph will proceed. Well, before that, let's look at the structures. So talking about alanine. So this is the structure of alanine. COOH, NH3 plus, H and CH3. Now what will happen is the first ionization will take place. This is the first species, and as you can see here, this is fully protonated. This species is fully protonated. Now what will happen is first ionization will take place. This is how the first ionization will take place, and the pK1 will be 2.3. The pK1 will be 2.3, and here we have a certain value of OH minus ion concentration. Now what we'll get here is, what we'll see here overall is this particular group, that is the alpha carboxylic acid group, will get ionized first. This will get ionized first, and in this state we can say that alanine has a net positive charge, that is a +1 charge. This is alanine with +1 charge. And also we can say that yes, net charge is +1. Net charge is +1. Moving forward, ionization has taken place. And what we can see here is COOH has now formed COO minus. And the net charge, this is because of pK1, and here the net charge was 0. Here the net charge is -1. The net charge here for carboxylic acid group is -1, since it is getting converted into carboxylate ion. And what it is forming is this particular species. It is forming this particular species where we have COO minus, we have H, we have NH2, and we have CH3. Sorry, this is NH3 plus, this is COO minus. Now what we can see here is a very important thing. Actually, if we try to encircle this one, This is a special species. This is a very special species which is commonly called as a Zwitter ion. Well, Zwitter means having both charges. Actually, this is Zwitter ion which behaves as a dipole because it has a positive charge as well as a negative charge, and because of this there is a net charge of 0. There is a net charge of 0, and so this is species number 2. This is we can say half protonated and half deprotonated. Now in this case the net charge is 0, so we can write here alanine 0. Now what we can say, that the pH at which this Zwitter ion exists, this is a special pH, this is a special value of pH at which Zwitter ion exists is called as the PI. And this PI is very important, as this is the isoelectric point. And why is it called as isoelectric point? Very important, because if we carry out electrophoretic mobility experiments, what we will see here is electrophoretic mobility of this particular species, of this Zwitter ion that has a net charge of 0. The electrophoretic mobility will be 0. And this at this PI is that value of pH at which this Zwitter ion exists, and also electrophoretic mobility is 0, and it is least soluble. At its PI, an amino acid is least soluble, so the solubility is the least in this state because of an overall net 0 charge. Now let's come to the next ionization. The next ionization choice is obviously this NH3 plus group. The second ionization will start to occur. Now there is pK2, and the value of pK2 is approximately 9.7, and here obviously the OH minus concentration is much greater. Obviously the pH is much greater, as you can see, and as a result this NH3 plus is also deprotonated, and what you finally get here is CO minus H, NH2 and CH3. And this particular species, that is species number 3, this is fully deprotonated. This is fully deprotonated, as you can see here, and what we can say is the net charge here is -1. You can calculate it very easily. This is -1. The net charge here is -1, and as a result the species we have here is alanine -1. This is the species that we are having here. So this is the overall story of alanine, the overall ionization of alanine, and if we try to represent it on this particular graph, what we can say here is this ionization is taking place in this way. Let's say first at a pH of around 2.3, this ionization took place. This is the value of pK1. This is pK1, and then this graph was rising, and at a pH of around 9.7 there is another ionization taking place, and then this is how this graph became. Well, now what we see here is at this point, approximately at this point, pK2 was there, and obviously as what you can comment from this is at this point, at this particular point, alanine 0 concentration will be equal to alanine -1 concentration. Similarly, at this point, alanine -1 alanine +1 concentration will be equal to alanine 0 concentration. And both these points, both these zones will be actually buffering zones. These are buffering zones. Similarly, this is also a buffering zone. These are buffering zones, right? Because you have a pK1, you have a pK, you have a pH, and obviously you had seen these graphs. You had seen these graphs for in the lecture of water, pH, and buffer. You had seen this for different like different acids. You had seen it for CS3COOH. You had also seen it for polyprotic acids. Like if there are polyprotic acids, like these are examples of polyprotic acids because there are more than one ionizing groups which are getting deprotonated. So yes, the graph is similar to the graph of carbonic acid as you can recall from the previous class. Well, so these are two buffering zones, and actually we find a PI here. We actually find a PI here, and this PI is this point, exactly in the middle of pK1 and pK2. Here we find the PI, and let's talk about the values. If we talk about the values for pK2, as you know, for pK2 it is 9.7, for pK1 it is 2.3. It is 2.3, and what we can say is before pK1 the dominant species here was species number 1. Then from pK1, after pK1 and before pK2, the dominant species here was species number 2, and after this pK2 the dominant species is species number 3. This is very important to note. And at the pK, actually it is half ionized. It is half ionized. Both are having equal amount of concentration. So because 50% ionization has taken place at each of these points, that is at pK2, at pK1. And this is how we get the PI. We get the PI, that is the isoelectric point, and in this case it is 6. But how do we calculate it? Well, I told you it's 6, but there is a way to calculate it. It's very easy. If you want to calculate PI for any, for example, if we are calculating it for alanine, or let's say for any amino acid with non-ionizable R group, for any amino acid with non-ionizable R group, what we can say here is PI is equal to pK1 plus pK2 by 2. It's average of both. It's average of both, and it's very simple. This is how we calculate it. So what we can say here is PI is, let's talk about alanine here. This is a general case, and for alanine the value of PI is actually pK1 plus pK2 by 2. What we can see here, pK1 is around 2.34, and this is around 9.7 divided by 2. So what we get here is approximately the value is 12. 12 by 2 is actually 6, and this is the value of your PI. This is how you calculate your PI. Now I have a few questions for you. Well, I'll be waiting for your answers in the comment section below. So this is your list of questions. Let me give you these few questions. Well, the question is now first I'll try to give an example. First, I'll try to explain with an example, and then I'll give you questions. Well, at pH less than 2.3, what do we observe? Obviously, the first species is dominant. The dominant species is alanine plus 1. This is the dominant species. Now, if we talk about a pH equal to 2.3, what can we say? We can say that there is an equilibrium between alanine plus 1 and alanine 0. There is they are having equal concentration. We can say that there is 50% ionization. 50% ionization. Also, if we talk about pH is equal to 9.7, at this point what we can see is this is exactly the pK2. This is exactly the pK2, and this was exactly the pK1. For this, the species alanine 0 and alanine minus 1 have equal concentration. That is, we can again talk about 50% ionization. So this is an important concept. Now, I'll ask you 3 questions. First, at what pH? At what pH? Question number 1. The predominant species the predominant species is alanine 0. No need to give a specific answer. You can also give a general answer like greater than, equal to, or lesser than, in that way. So this is your first question. Then the next question is half of alpha amino groups are ionized. Half of alpha amino groups are ionized. At what pH? And the third one, average net charge is plus half. At which pH? At what pH? So this is your third question. So please answer all these three questions in the comment section down below. So I'll be waiting for your replies in the comments. Do let me know the answer in the comments. Fine. So let's move forward. Let's now talk about the titration of acidic amino acids. Now, next we'll be talking about the titration of acidic amino acids. So now, if we talk about acidic amino acids, well, we can talk about aspartate, we can talk about glutamate. If we want to consider glutamate, for example, glutamate is E. We'll try to draw the structure first. This is COOH. This is NH3 plus. Then we have H, CH2, CH2, COOH. This is obviously, this is your alpha carbon. This is beta carbon, gamma carbon. I hope you can see the structure. So this is the structure of glutamate. As you can see here, this is the alpha carbon, beta carbon, gamma carbon, and to this gamma carbon we have this carboxylic acid group attached, and therefore it is called as a gamma carboxylic acid. So for this gamma carboxylic acid, if we try to look at the pKr, the value of pKr here is 4.25. Well, we know it that the pK1, approximately the pK1 here will be 2.19, and the pK2 here, the pK2 in this case, will be equal to 9.67. So this is the value of pK1 and this is the value of pK2. Now, there are other acidic amino acids as well. Like for example, if I tell you about this phenolic group or sulfhydryl group, like there is Y and C as well. If you talk about Y is tyrosine, and for tyrosine, the group is, there is a carbon, then there is a benzene ring, then there is this benzene ring, and there is this OH. This is the phenolic group which is attached. Similarly to cysteine, we have this CH2 group, and there is this sulfhydryl group. For aspartate, we have CH2 and COOH, and for glutamate, as we can see, it's CH2 and COOH. This is the, obviously this is the beta carboxylic acid group, and this is the beta gamma. This is the gamma carboxylic acid group. So these are the major amino acids which will give H plus ions, and these have acidic amino acid groups. Especially we will talk about these two. And in the end we will be talking about Y and C. So what we are concerned about, this graph basically corresponds to glutamate and aspartate. Now let's talk about the ionization of glutamate. Let's look at the structure of glutamate. COOH, then there is NH3 plus, H, there are two carbons, and then there is COOH. This is the fully protonated form. Species number 1. Then what is happening? There is this ionization taking place. And the first ionization will take place obviously from here. So the pK1 here will be 2.19. Now it's very easy. You've already seen it for the for non-polar amino acids, that is with amino acids having having a side chain which is non-ionizable. Now it's very simple that the first group to be ionized will be the alpha carboxylic acid group. Now, which will be the second group? Will it be NH3 plus or will it be COOH? Obviously, it will be the R group COOH, and this will be the second group. So we'll see that. But as of now, we are increasing the OH minus ion concentration. This ionization of COOH, that is the alpha carboxylic acid group, is taking place. And then the structure that we are getting, is here we are having COO minus. Then here we have 2 carbons, and here we have COOH. Now the next target is this one. This will be the second group to be ionized. Now here what we see is there is this pKr. The pKr value, as you have seen, is 4.25. This is also getting ionized, and as a result the structure that is being formed is COO minus H, NH3 plus, carbon and COO minus. Now the last group to be ionized is this one, and for this we'll have pK2, and pK2 will be equal to 9.67, and the final species that we get is COO minus. Here we have H, NH2, carbon and COO minus. Now let's look at the net charge. The net charge is also this. Let's give the species number. This is species number 2, this is 1, this is species number 3, and this is the 4th species. Now let's talk about the net charge. The net charge here is +1. As you can see, you can easily calculate it. It is +1 here, it is 0 here. So from +1 it is going to 0, then it is going to -1, and then finally it is going to -2. +1, 0, -1, -2. This is for this is common for all acidic amino acids. Here it is, we can write it as glutamate with +1 charge. Then we can write glutamate with 0 charge. We can write glutamate with -1 charge, and here we can write glutamate with -2 charge. So this is the net charge. And now the important thing to note here is, well, in the previous case as well, when we were taking the pi value, actually we were considering 2 species. The species that is towards we were considering the pKs that were towards the right and left of the Zwitter ion, that is with respect to the species which has a net charge of 0. We were looking at the left hand side and the right hand side equilibrium, and we were taking into consideration the pKs of both these equilibria, and then we were taking the average of these two. Well, here we'll be doing the same as well. We'll be marking this particular species, and we will consider this pK and this pK, the right and left of 0. So this will be considered here. So if we want to calculate the pi for acidic amino acids, obviously they are charged. So it will be equal to pK1 plus pKr. pK1 plus pKr, and pKr is obviously of the side chain carboxylic acid group, and we will be taking an average of these two. So this is how we will get the pi. This is how you can calculate the pi for charged amino acids, and we will take the closest values. We'll take the closest values like pK1 and pKr, because pK2 is far away, so we won't take that. So if we want to calculate the pi for glutamate, it will be 2.19 plus 4.25 divided by 2. That gives you a pi value of 3.22, and 3.22 will be the value of pi for glutamate. Now let's look at the titration. Let's look at the graph ones. Well, here what will happen is, first this ionization will be taking place here at around 2.19. This ionization took place. Now what will happen is, there will be another ionization there will be another ionization here around 4.25. Then there will be another rise here, and then there will be a final ionization. There will be a final ionization that will take place at around 9.67, like this, and this will rise like this. This will rise like this. So this will be your pK2, this will be your pKr, and this will be pK1. This is pK1. So the values, you know it very clearly. This is pK1 is 2.19, pKr is 4.25, and pK2 is 9.67. These are the values. And here, how can you actually calculate the PI? You can calculate the PI from here. This will be the midpoint of these two. This will be the midpoint of these two, and you will be calculating it like this. You'll be calculating it like this. This will be the PI, and the PI, you can calculate it. It is 3. This is your PI, and the value of PI here is 3.22. This is for glutamate. This is for glutamate, that is for acidic amino acids. You can also do it for aspartate. It's as simple as that. Now let's finally talk about the titration of basic amino acids. Titration of basic amino acids. Now, which are the basic amino acids? Well, you know it very well that it's arginine, lysine, and histidine. Arginine, lysine, and histidine. Let me tell you about this lysine, and let me tell you about arginine. For arginine, the pKr value is 12.5, and for lysine, the pKr value is 10.5. So it's 10.5 for lysine, and for arginine it's 12.5. For histidine, the pKr is actually very less, and it is even lesser than the pK2, so it's 6.0. 6.0. We'll look at all the pKrs once, but let's just write it for these three amino acids. Now, if we talk about histidine, I won't draw the whole structure here, as the structure I have already taught you about the structures. We'll just show the side chain here, because that is what we are more concerned about. The other parts are common to all amino acids. So here it's CH2, and here it's like this. There is NH. Here we have N plus. This is the shield-like structure of histidine. That is the imidazole ring. This is the imidazole ring, and for this imidazole ring, we can see that the pKr is 6.0. It's 6.0, right? So we'll draw the curve for histidine. We'll draw the curve for histidine, but we'll also look at the ionization of lysine and arginine in a very crude manner. We won't be drawing the whole structure, rather we'll just look at the functional groups. So what we can see here is, if you draw the general structure here, this is H, this is COOH. Obviously there will be pK1 value here will be 1.82. It varies from amino acids to amino acids. This is NH3 plus, and here the pK2 will be 9.17, and here we have histidine, and histidine has this group CH2. Obviously let's draw it, since we have already drawn this much, so we can draw this as well. NH, and here we have N positive. This is the imidazole ring, and this has a pKr of 6.0. Now let's look at the structures. Let's look at the structures for histidine, simply for histidine. Well, if we have histidine here, we have COOH, we have NH3 plus, that is the fully protonated state, H, and here we are having histidine. Histidine has this group of NH3 plus. Simply, we'll just look at NH3 plus as a group, as a side chain, as a basic group. So what we see here is the first ionization that will be taking place is of this one. This is the first ionization. Then we'll write the pK1. pK1 is 1.82. Now what happens is, obviously at this particular pH, the COOH will get ionized. Then we'll get another species. We'll get species number 2. And what is species number 2? COO minus NH3 plus and the side chain of histidine NH3 plus. This is the structure. Now what will happen? The second target is this one. This is the second target. So what will happen is another ionization will take place, and the pKr for histidine is 6.0, and at this particular pH it will get ionized. COO minus NH3 plus H, and histidine will now have NH2 group, and there is another ionization taking place. This is the last ionization, obviously in the alpha. This will be for the alpha amino group, and what we get here is pK2. The value of pK2 here will be 9.17, and what we'll get here is CO minus, we'll get H here, then NH2, then for histidine also we'll have NH2. Now let's calculate the net charges. So this is species number 3, this is species number 4. What we can say is the net charge here for histidine, this is net charge here is +2. Then here it is histidine +1. Then it is histidine 0, and then we have histidine -1. So what we see here is from +2 it is going to +1, then it is going to 0, and finally it is going to -1. So +2, +1, 0, -1. This is the order. And obviously for calculating the pK, for calculating the PI, what we will consider is the left and right equilibria, left and right equilibria of this species with net charge 0. So we'll be considering pKr and pK2 for histidine. Now let's consider the case of arginine and lysine as well. Arginine and lysine, we know that the pKr here is 12.5 and pKr here is 10.5, right? Now let's talk about lysine. So lysine will have a net charge of +2, then it will have pK1. Obviously the carboxylic acid will get ionized, it will get deprotonated, then we get lysine +1. Then again there will be this pK2 happening, because the value of pK2 here will be lesser than that of pKr. The pKr has a higher value here, so the first ionization that will be taking place relatively here, that will be of the alpha amino group. This will be valid for both lysine and arginine. It will be exactly similar for arginine. For arginine it will be exactly similar, this exact ionization, just the values will change. Now there will be lysine 0, there will be pKr, and finally lysine will get converted to lysine -1. Well, what we see, the species are the same, like the net charges are the same, +2, +1, 0, and then -1. But for histidine, what we see is that pKr, this particular equilibrium occurs before pK2. Whereas for others, it is pK2 first and then pKr. Like for lysine and arginine, it's pK2 first and then pKr. This is very important. Now, the same case is for arginine, and what we see here is, yes, the net charge is +2, +1, 0, -1, and obviously what we consider here is this and this, and finally if we calculate the value of PI for basic amino acids. So for basic amino acids which are obviously charged, what we see is for PI, what we'll write here is pK2 plus pKr. So this will be same for histidine as well. See, for histidine we were taking pKr first and then pK2, but the order doesn't matter if you are taking average. So pK2 plus pKr, and where the pKr here is NH3 plus. Just because of the NH3 plus group divided by 2, and we get this as the formula for PI calculation for basic amino acids that are charged. And now, for example, for histidine, if we calculate the PI, this will be 9.17 plus 6.0 by 2, that is 15.17 by 2, that is approximately 7.58. That is 7.58, and this is how we calculate the PI for histidine. Now let's plot the graph. Let's plot the graph for histidine as well. Well, what happens here is this ionization is taking place. there's this first ionization occurring here. This is pK1. This is the pK1. Then there is this rise. Just a moment. Then there is this rise here. And we are coming to this pKr. Then we are coming to pKr, that is at a value of 6. Now what will happen is this will again rise, and at a value of 9.17 there will be pK2. There will be pK2, and this will rise again, and this will go here. So this is how we see this is pK2, this is pKr, and this is pK1. Now for PI calculation, what we saw, the value of PI here is 7.58. So at this point we get, so this is the midpoint of this two, and at this particular point we get the PI. This is the point where we'll find the PI, and here for histidine the PI is 7.58. This is the PI for histidine, and obviously the values of pK1 and pKr, we already know it. This is 1.82, this is 6.0, and this is 9.17, and finally this is how graphically we can see the PI. So we have learned the titration of all types of amino acids for amino acids with no ionizable groups, like for all the non-polar and most of the polar uncharged amino acids. Then we have seen it for acidic amino acids, for charged amino acids, acidic as well as basic. Now let's summarize the overall values in a very general sense, and then we will be calculating the charge on a peptide. So these are a few values which I have written. Well, the pK1 for alpha carboxylic acids, if we consider almost all amino acids and we actually look at their values, we can take an average. And for calculations in general, if it is not specified, in most of the cases, if it is asked in examinations, it will be specified. Like for the alpha carboxylic acid group, it will be specified the value of pK. Similarly, for alpha amino group, it will be specified. But if it is not specified, for a safe assumption, what you can do is you can take the value as 2.2, around 2.2 for alpha carboxylic acid group, and for alpha amino group you can take the value as the value of pK2 as 9.5. Now for pKr, this is very important. For pKr, if we have acidic amino acids, that is negatively charged amino acids, for aspartate the value is 3.65. The value is 3.65. That is for the beta carboxylic acid group. Similarly, for E, that is glutamate, it is 4.25. So for glutamate it is greater, and for aspartate it is less. Now coming to basic amino acids, that is the positively charged amino acids. First, for arginine. Arginine has this guanidino group, and this guanidino group has the highest value of pK. It has the highest value of pK among all side chains, and the value is approximately 12.5. So for lysine, if you look at lysine, the group here is the epsilon amino group, and for this epsilon amino group the approximate value is 10.5. So for arginine it's 12.5, for lysine it's 10.5, and for histidine it's 6. As simple as that, right? So for histidine, for the imidazole group, as we have already seen, it's 6.0. Now for polar uncharged amino acids, I had told you about cysteine and tyrosine. Well, the group here is, for cysteine it's sulfitrile, and for tyrosine it's phenol. Now this phenolic group, this has this side chain pK, that is pKr of 10.07, and for cysteine it's 8.18. Well, for cysteine, if cysteine is asked, these are two kind of exceptional cases. So for cysteine, for PI calculation, you will simply take pK1 plus pKr by 2, and if you actually calculate it, if you take the average of pK1 and pKr, and you divide by 2, you add and divide by 2, the value you will get is of 5.07. Approximately it is 5.07, practically calculated value 5.07. Now if you talk about tyrosine, for tyrosine it's 10.07, and if you calculate the PI for here, you will for this you will simply take the pK1, you will take the pK2, and you will take the average. Here this is not considered, this is simply 5.66. So please remember this for cysteine and tyrosine. Well, but for most of the cases, for most of the questions, 90% of the questions, for 90% of the questions, you will take these values. You will be requiring the values for acidic and basic amino acids only, or else it will be asked for neutral amino acids, and it will be very easy. You will just take pK1, pK2, and you will take the average of these two for PI calculation. Now, there is an important concept. There is an important concept of calculation of charge. Now, you have already seen the concept of calculation of charge. We will now talk about calculation of charge on peptides. What are peptides? Obviously, peptides are the polymers of amino acids. Amino acids, when they get joined together and they get bonded by peptide bonds, then the structures formed are called peptides. Peptides have these peptide bonds in between, and in the extreme ends, in the extreme left end, they will be having an amino group that will be called as the N terminus, and in the extreme right they will have a carboxylic acid group that will be called as the C terminus. So this is the basic idea of a peptide. It is nothing but a polymer. So this is how amino acids join to form a peptide. Amino acids join to form a peptide. Now, the main concept here is to calculate charge on a peptide. The second topic, as you can see here. Well, it's very easy. Actually, these foundations were very important to understand charge on a peptide. Now it's very easy. Now you can understand this in the form of ionization. Well, if the value is 2.2, like if I'm asking you what is the state of this carboxylic acid group at pH equal to 4, so I'll simply say that, well, the species that will be dominant will be COO minus. So for this, the net charge will be -1. Earlier it was 0, now it is -1. So this is how we can calculate the net charge on a peptide depending on whatever groups it is having. And also the amino acids won't be free, only except the extreme left and extreme right end, no other group will be free. There is no free amino or free carboxylic acid group in between. So there you will be considering only and only the side chains, and for side chains obviously we'll take into consideration the acidic and basic side chains. Now, without further delay, let's come to the point. Let's finally come to calculation of charge on a peptide. So we will actually address this concept through a question, and this question is calculate charge on the peptide at pH equal to 7. This is your particular question. This is your question. Calculate pH on the peptide at pH equal to 7. Now I'll give you a peptide. Well. The surprise here is the peptide is peptide itself. So it's proline, glutamate, proline, threonine, isoleucine, aspartate, glutamate, and then finally, as I told you, there will be this free amino group here since it is not bonded to anyone, and here we'll have a free carboxylic acid group like this. Now, as we know, this is your alpha amino group, and for alpha amino group, what we can consider, it's very important to specify the groups and their pKs, because that is very important. Now only then you can be able to calculate the charge at a particular pH. So here we take into consideration pK2. Let's consider it as 9.5 on an average. For COH, let's consider it as pK1 as 2.2. Fine. Now at pH 7, obviously the one which had a pK less than 7, if the pK is less than 7, all those groups will be deprotonated. Obviously here NH3 plus group won't be deprotonated because here the pH is 7. It hasn't reached the cutoff yet. And for pK1, it has already crossed the cutoff. Now let's search for any kind of side chains here. Well, here you can see a side chain. You have COOH. We are talking about a fully protonated state here. Then we will carry out the deprotonation. Here what we can see is for this aspartic acid, again, there is COH. Here we are having COH. These are the only groups that are charged. Otherwise, no other groups are actually capable of ionization. Now let's talk about so this has a pKr. This has a pKr of 4.25. Similarly, this has a pKr of 4.25, and this has a pKr of 3.65. This has a pKr of 3.65. Now at pH 7, this is maintained. This is not ionized. This is ionized. This is ionized. This is ionized, and this is ionized. If this is ionized, obviously, if this is ionized, the group now we have is COO minus. The group here we have is COO minus, and the group here we have is also COO minus. This group here is also COO minus. Earlier, the net charge we had initially, this was +1 here, then we had 0 here, 0 here, 0 here, and 0 here. So the net charge at pH 0 was +1. But here, since this group, this lost its H+, this lost its H+, this and this, all these lost their respective H+ ions, and they got ionized, and they are now having a net charge of -1. So it's -1 here, -1 here, -1 here, -1 here, and +1 here, since this did not get ionized. So it's +1 here, -1 here, -1 here, -1 here, and -1 here. So if you add all these, well, to calculate the net charge will be given by +1, -1, -1, -1, and this is how you get the net charge value as -3. And this is the net charge on this particular peptide, which is obviously named as peptide as well, at pH 7. Now I have a question for you. The question is, at pH is equal to 10, what will be net charge on this same peptide? Please give your answers in the comments section down below. I'll be waiting for your comments. So yes, that's all for this lecture, that is lecture 6, amino acid titration and charge on a peptide. I hope you are enjoying this series on biochemistry, that is biochemistry decrypted, and all these concepts are gradually getting cleared. Well, let me know your feedback about this particular course that is going on in the comment section below. Also, if you need any other kind of help, any other kind of improvement you want to see in these videos, do give the suggestions. Well, all the channels and groups are mentioned here. If you want to download the notes, you can download you are having the link in the description. Everything is given to you. Well, now what I want to request is, please share these videos, because these videos are completely free of cost and they are of top-notch quality. I am trying my level best to maintain the top-notch quality in terms of content, in terms of visuals. Please share this with all your friends, whoever is not having access to premium resources. So please share this so that they can also prepare for their exams, or maybe just learn this subject for the love of this subject. So yes, thank you so much for watching. Do share this video, do leave a like, comment, and yes, if you want, you can subscribe to this channel to get regular notifications. So thank you so much for watching. See you in the next video, that will be on peptide bonds, peptides, and polypeptides. Thank you for watching. See you in the next video. Bye everyone.